Tibet, sí. No, we are not seeing the screen yet.
You're not? That's weird. But I just, okay. Okay, because I got a message that we're being live streamed and it didn't. All right, so now it's going to share the screen. Yes. Okay, so you should see a blue thing, and now you should see the real thing.
Yeah, that's perfect. Okay, great. Well, hi, good afternoon to everyone, and thank you very much for joining us today. This is the last seminar before the holidays, and I think it's a fantastic way to close the semester with today's speaker. So Professor Argyris Papantonis studied biology at the National and Kapodistrian University of Athens, where he also undertook his PhD studies. Then in 2008, he moved to Oxford for his postdoctoral work at the Sir William Dunn School of Pathology with Professor Peter Cook, we were just discussing. And in 2013, he was appointed as a junior research group leader at the Center for Molecular Medicine in Cologne. And in 2018, he was recruited to the medical faculty of the George August University of Göttingen, where he currently holds the chair for translational epigenetics. So we are, oh, sorry, that's my daughter on the background. So we are delighted to have you here and very excited to hear about your recent work. So take it away. Thanks.
Well, thank you, Myra, for the invitation. It's really great. I actually told my kids, I've been traveling a lot the last couple of months, and I said, well, you know, Friday I'm giving a talk in Mexico, and they were both absolutely annoyed. They were like, what, you're going to Mexico without us? This is unacceptable. Then they realized it was Zoom, so it was slightly better. So what I'm going to talk about something today that is unpublished, but you know, we always talk openly about our stuff in my lab. And what we generally study is this relationship between the spatial organization of chromosomes and their function. And of course, in order to study a relationship like this, you need to find ways to understand it. So the understanding that we had of this relationship up until recently was, you know, what we call the Michael Jordan conundrum. And this means that, you know, there's this textbook, 2 meters of DNA if you unravel all your chromosomes, and how do you fit them in a very small micrometer long nucleus. But if you really think about it, up until very few years ago, our appreciation of nuclear organization was this picture, this very famous picture from the Kramer brothers. So essentially the fact that individual chromosomes occupy their own distinct territories within the nucleus. And further in from that, we only had some, you know, specific low throughput DNA fish studies, right, where they told us about individual loci and where they might land relative to one another. So now that we have molecular tools, and I'm going to talk about them, although I'm sure you have heard Myra talk about them quite a bit, we need to also find ways to meaningfully perturb this structure to function relationship in order to understand it. And the way we are doing it, one of the ways by which we're doing it, is by using cellular aging as a tool. And this idea developed many years ago when I first started my lab in Cologne, because Cologne is a hub for aging research. And just by looking at literature back then, you could see that if one looks down the microscope, you can see really large changes in genome organization when you look at cells that have aged in different contexts. For example, and this is actually work that comes from Peter Fraser and Wolf Reich's lab in Cambridge. If you have an oncogene-induced senescence stimulus inside the fibroblast, you have the formation of this very large heterochromatic foci. And if you look at premature aging syndromes like the HUBS progeria, you have again these very sort of dense heterochromatic foci. So one would naively think that these contexts would give you such large changes in 3D genome organization that they're worth studying at high resolution. That's what we thought at the time. I should say now that we were pretty wrong of what one can see or cannot see at the molecular level, but that was the idea. So we wanted to study replicative senescence. This is the senescence that is normally registered into the program of every proliferating cell in our bodies, and usually comes about as a result of telomere shortening over time. So every cell will face that moment where the telomeres will be short enough that it will not be able to divide anymore. And early on in life, this type of senescence and other types of senescence as well, they can be very, very beneficial. So for example, these types of senescence can be used for development. So for example, there's tissue between our fingers as we develop, and this is being eliminated by senescing cells. It's very good for wound healing, injury repair, and of course, whenever a cell sort of aberrantly gets stimulated by an oncogene, you can actually drive it into senescence and make sure that the tumor doesn't fall. But late in life, you can have what is now understood as chronic senescence. So this is the accumulation without clearance by the immune system of a large number of senescent cells in different tissues of the body. And these cells tend to secrete lots of pro-inflammatory and stress factors into their surroundings, which of course gives rise to chronic inflammation, which is now understood to be a very important contributor to organism aging and age-related disease. And there's very recent work wherein clearing these senescent cells from tissue actually leads to much healthier tissue, longer lifespan in mice, less occurrence of cancer, and so on and so forth. So there's some merit in taking away the senescent cells. So what we hypothesized was rather simple. We said, okay, what if the aging of a given cell, that is generally a stochastic process and it occurs at different times for different cells, is driven by early and deterministic events at the level of chromatin. So what if we have chromosomal aging before you have cell aging? If this is the case, then one could identify these events by looking at how chromatin changes and how its organization changes. And because this will be through some particular factors, one could target these, reverse aging, or even stop it. Probably not, but delay it severely, I would say. So that was the sort of naive approach we took to this. And again, back then, what we did is we took 3 very different cell types that are developmentally very distinct, and we simply did an RNA-seq on them to try and find commonly deregulated genes as a result of these cells entering senescence. So we had mesenchymal stroma cells, we had fetal lung fibroblasts and umbilical cord endothelial cells from different donors, and we did RNA-seq, and there were 200 or more genes that were commonly and strongly downregulated when these cells committed to the fate of senescence. And if you look at the kinds of genes that you're dealing with, of course, you have all the expected stuff because these cells don't divide anymore. So the cell cycle is impaired, replication, DNA metabolism, so on and so forth. But there was this little yellow cluster up here that had to do with the conformation of DNA in chromatin, its assembly, its packaging, and so on and so forth. And these are just some of the proteins, the genes that encoded relevant proteins that were part of this cluster. And you can see multiple ones here. So there's condensing subunits, there's chromatin remodelers, there's a heterochromatic deposition factor here. And at the very top of the list stood 2 high mobility group proteins, which are going to be the center of my talk from now on. And why is that? This is because we found out that you could actually identify a senescent nucleus just by looking at whether these proteins are there or not. And this is an example from HMGB2. It's the same for HMGB1. The senescent nuclei that tend to be larger, like these ones here, are clearly the ones marked by the loss, the very, very severe loss of HMGB2. So why is that surprising? Well, this is surprising if you think about numbers for 2 reasons. One is that the high mobility group box proteins, as they're best known, they are very, very abundant. They're essentially expressed in most tissues and organs at decently high levels. B1 slightly more than B2, also because B2 tends to be a bit more earlier embryonic in development. They're very much alike, so they share more than 80% of their amino acid sequence, and they are the most abundant proteins in mammalian nuclei after histones. So for example, for HMGB1, we have about 1 molecule for every 10 nucleosomes. So we're talking about an incredible number, which you can actually appreciate by looking at these super resolution images of them being dispersed in the nucleus. So a senescent cell will go from hundreds of thousands of copies, if not more, to almost nothing within very few passages. So does this, offloading of such a huge protein concentration make a difference? So we started to ask, what does high mobility group box protein 2 does, right? What does it do? And so HMGB2, and I'm going to go quickly through this because this has been published. So HMGB2, when we combine Hi-C, which is a molecular method for looking at 3D genome organization, and ChIP-seq for finding positions that are bound by HMGB2, this is what we found. We found that it is strongly enriched at the boundaries of these very famous 3D chromatin domain called TADs, or topologically associating domains. And these boundaries are very important because they insulate each TAD from the next. And of course, if you don't have that, then you can have aberrant interactions across TAD that can lead to disease, including cancer, for example. So this insulation by HMGB2 was comparable to that we saw by CTCF. And as you can see here, it was held true in all the cell types that we looked in. So these are the fibroblasts, these are the endothelial cells. And of course, when cells are driven into senescence, then this demarcation is lost because, of course, you don't have the protein there anymore. You don't see a complete loss here because, of course, the population is heterogeneous. Each cell will go into senescence at its own pace. Now, this is all fine and good, but this is, senescence is the result of multiple things. So what we did is we took young cells and simply knocked down high mobility group B2. And the results were really astonishing. So just knocking down a single factor does 3 things. First of all, if you knock it down, both HUVEX and IMA90s have a reduced ability to produce nascent transcripts, so to really transcribe genes, genome-wide, between 15 and 30%. It's a huge drop, again, but just by removing a single transcription factor. The other thing that happens by removing HMGB2, you can see the knockdown efficiency here, is you have an increase in HP on alpha marked chromatin. So this is the deep constitutive heterochromatin that's usually in the periphery. And you have a decrease in the more plastic facultative heterochromatin marked by K27 trimethylation. And I should say that, by the way, these 2 changes, so the loss of transcriptional output and the shift in the identity of heterochromatin, is exactly what happens in senescence. So just by knocking down a single protein, you can phenocopy the 2 major chromatin-based changes that you see upon senescence commitment. And the third one is something that no one had seen before. And the reason, to be absolutely honest, was that we also found it by mistake. So we were just staining with antibodies that we had in our hands. And this was essentially going to be a control experiment. And this is what we saw. So this is a control pair of cells. You see HMGB2 everywhere. You see CTCF essentially everywhere in the nucleoplasm except the nucleolus. And this is the knockdown. No HMGB2. And all of a sudden, CTCF starts forming these large clusters. And this was exactly what one sees in the very large nuclei, large because also lamins are deregulated in senescence, that are senescent. And you can see these very distinct foci formed by CTCF. So this was a big surprise to us. And the next thing we did is we thought, okay, could this be aggregates like tau, for example, in neurodegenerative disease? And we washed out the nucleoplasm. Oh, there's a question. I'm happy to answer a question mid-talk.
Yes, I'm sorry. I was wondering that in your images, I can see that the high mobility group protein seems to be, the very few amount that is remained, is it enriched in the nucleolus?
Yes, I was about to comment on that. Yes, no, it's a very good point. The nucleolar staining of high mobility group proteins is a false positive result. And this is because, when you do live cell imaging of these proteins, what happens is when you add formaldehyde to cross-link them to chromatin, they immediately move away from that. And then somehow they get stuck in the nucleolus and you see them there. So this is the result of using formaldehyde cross-linking. And this is the reason why no one up until this point had ever chipped HMGB2 and HMGB1, because it was impossible with formaldehyde to capture them on chromatin. So if you do the proper way, which is to add a fixed length cross-linker first at low temperature, so at 4 degrees, and then stabilize that with formaldehyde and then stain, you don't get staining in the nucleolus anymore. But for the purpose,
a technical question. Then, if you fix the cells with methanol, for example,
we've tried every single way, and none of them work well enough. The only one is this combination of a fixed length cross-linker like EGS or DSG, and then formaldehyde, and it has to be at 4 degrees to really reduce the on-off motion on chromatin. And for all the IFs that we've done, we stick to the sort of simple 4% formaldehyde, just because it's simple, because we don't care about the nucleolar staining. It doesn't affect our readouts, but the proper picture would be without nucleolar stain. That is absolutely true. Now, sure. So now what I was trying to say is that we were worried that these are just aggregates and they have nothing to do with chromatin. So we extracted the diffuse pool from these cells, then fixed by, permeabilizing the nuclei, washed stuff out, then fixed, and still, these were left behind. So to cut a very long story short, we could re-express HMGB2 in these cells, and this is tied with GFP. And when we did this, we can actually, for most of the cases, and this will become apparent later, we can rescue the formation of these 6. So it is enough to put HMGB2 back to not allow for these CTCF clusters to happen. So what we had in our hands was a new presumably structural hallmark of senescence that we called senescence-induced CTCF clusters, or 6. And I think you can see from these steady images here, the super resolution images, that in a young cell, the B2 and the CTCF do not really colocalize. They sort of interdigitate like this. And then when you lose B2, you have the clustering of CTCF in these very large entities that seem to contain many tens of molecules. And to add a 3D sort of flavor to that, because again, this was all published in 2018, to add a 3D flavor to that, we did some high chip, because back then it was difficult to get high resolution, high C, to look at what happens to loops. And what we found was that we had longer CTCF loops in senescence, and slightly more CTCF loops as well in senescence. And these used to be across positions previously bound by HMGB2. So you can see this very long loop that comes across this position, and you can see this very long loop, for example, that loop that come across these positions that were previously occupied. So the idea we had at the time was, okay, what happens is you make these longer loops and they all come together. You have these packs of loops coming together and somehow reorganizing the genome. That's the idea. But we didn't know 2 very important things. One is how, so what is the molecular mechanism that allows this protein that has nothing to do with CTCF, right? If you pull down CTCF or HMGB2, you don't get HMGB2 or CTCF. They don't interact directly. And they don't bind the same positions in the genome. If anything, they can be a few kb apart, but really they don't compete for anything. So how does removing this one here allow these long loops to happen? To address this how, we started off by doing lots of co-immunofluorescence stainings, right? And we took all the histone marks that you can imagine, and we tried to, we'll find the type of chromatin that these happen in. And we couldn't get any colocalization. Now, one other problem that we have, and I'll get back to the colocalization issue, one other problem that we have is that each cell sort of senesces at its own pace. So you're always battling with heterogeneity. So we have now come up, and actually I've finally managed to also, finish the paper. So it will be on the bioarchive next week, hopefully. We've devised a way of inducing senescence very quickly and very homogeneously in a cell population. And we do this by repurposing a small molecule inhibitor called ICM that was supposed to do a very different thing, and it's described in this paper here, and I'm happy to discuss it afterwards. But the bottom line is that if you hit IMA90s, which is the most well-known model of cellular senescence, if you hit them with ICM for up to 6 days, you can get them to stain positive for beta-gal activity, which is one of the markers of senescence, to stop dividing, and very importantly, to form this beautiful CTCF foci that you can see here. And of course, have everything else, right? You lose HMGBs, you turn on P21 to have cell cycle arrest, and you start switching the identity of heterochromatin. But again, this really beautiful large CTCF foci. And if you do a gene expression profile and you correlate this to replicative senescence, they correlate very well, but it doesn't really correlate to oncogene senescence. So it's all nice and good. It seems like within very few days, we can actually induce a senescent-like phenotype that is very much like the type of senescence that we want to study. So what happens now? Well, you can treat with this drug for, a decently long time. If you treat for up to 6 days, 4, 5, 3, 4, 5, you get more than half of your cells having these very nice CTCF clusters that you see here. This is your DAPI staining. There's no HMGB2 because the cells are senescent. And if you keep treating for longer, you get even more cells that have these very pronounced foci. Now, these look very round. And being in the era of phase separation of everything, we thought, these look like blobs. Could this be true? So we hit these cells for just a minute with 1.6 hexandiol, which is a rather nasty chemical that is, however, known to alleviate the interaction that can give rise to these blobs. And indeed, if you treat cells treated for the short amount of time with hexandiol, you can completely dissolve the formation of these clusters. And as you can see, these are senescent cells because they don't have an HMGB2. But if you do this in the long-term treated cells, these don't care. The clusters persist almost in every cell, suggesting that whatever the nature is, early on when it's happening, it's susceptible to this chemical, but later on it is not. And this, to me, was also telling of the experiment where we re-expressed HMGB2, where we could rescue most of the foci formation, but not all of them, which meant perhaps that some of them have transitioned in this other biophysical state that is no longer susceptible. And although we've done this experiment a long time ago, just last year, there was a publication in Drosophila and one in human cells showing that the extended DNA binding domain and the N-terminal domain of CTCF, they have the propensity of forming blobs inside of human cells. And these blobs were, to some extent, discernible by hexandiol, just like the ones we have. So, okay, this is all nice and good. These are likely phase-separated entities. Okay, still, we don't know what makes these foci emerge. So what we did is we did a super simple experiment. We pulled out CTCF and did a co-IP mass spectrometry blind test, essentially, in young cells and in senescent cells. And we just compared the two interactomes and said, do we see any change? And there were 13 factors that were significantly enriched in the CTCF interactome in senescence. Of these 13, we have tested 7, and only 2 have an effect in the formation of these senescence-induced clusters. The first one was BANF. Now, BANF is very interesting. So BANF, ignore panel B for a second, and we're going to focus on this schematic here. So this comes from a Cell paper from David Gurley's lab, where they show that during division, when the nuclear envelope is destroyed, BANF, or BAF, as it is also known, will coat the mitotic chromosomes to keep them together in a bundle as they migrate towards the new nucleus before the nuclear envelope forms. And then as the nuclear envelope forms, they will act as a molecular glue connecting the envelope to the key anchor proteins on the inside of the nuclear envelope, like emerin, for example. So this is a very small protein. I think it's about 25 kilodaltons. And you can do these plots asking whether it's disordered, because the disordered structure of a protein or disordered domains of a protein can help in phase separation. And as you can see, with perhaps the exception of very few amino acids here, this is a very well-ordered protein. If it has a value under 0.5, it's very well ordered. And as a sort of control next to it, you can see CTCF, which is far larger. It has this ordered middle part, which is the DNA binding domain. You see the 11 zinc fingers here, one after the other, very well ordered. And then you see a disordered C-terminal region and some small N-terminal disorder regions, just as a comparison. Okay. So now, if we look at where BANF is, it's embedded in chromatin, both in young cells, but also in senescent cells. And then if you do a knockdown of BANF, you cannot form the CTCF clusters anymore. So it's enough to remove BANF to not have the ability to form CTCF clusters. The second protein that had an effect on the formation of CTCF clusters was SRM2. And SRM2 is a completely different thing. So this is a key component, key structure, so it's a splicing factor, an RNA binding domain, an RNA binding protein, and a key structural component of nuclear speckles. And nuclear speckles are these membraneless, absolutely phase-separated entities that are in the nucleoplasm. And most people think that you can store lots of transcripts there, lots of splicing factors. They're considered to be at least partially active as regards splicing. Now, if you look at this SRM2, it's very, very large. It's about 350 kilodaltons in size, and it's mostly unstructured, as is the case for most large RNA binding proteins. So it has this huge, very long domains that are completely unstructured and rich for the kind of amino acids that you would expect. Again, this is the comparison to far smaller CTCF. And they mark speckles. So when we did, this is the first co-IF that we do that has any sort of colocalization. When you co-IF for CTCF and SC35, which is the key marker for speckles, which the lab of Tuksha Akhtas has shown that this actually antibody is not what we thought it was. It's an antibody that actually recognizes SRM2. So when you do that, you have a perfect colocalization of the CTCF clusters and the speckles. And in fact, you can find speckles that don't have a CTCF cluster underneath. Here's one here in red that hasn't got a cluster under it, but you can't find a CTCF cluster that is not at a speckle. And if you knock down SRM2, the speckles mostly dissolve, and of course you can't form any type of CTCF cluster. You completely abolish their formation. So if this is true, it would mean that if we're able to manipulate nuclear speckles, we would be able to manipulate these CTCF clusters. So we can use tautomycin, which is a protein that disperses, a drug that disperses speckles. And this is what we do here. You can see the dispersal. And if you do now this in ICM treated cells, you see very nicely that, the CTCF nicely follows the dispersion of the speckles. In some cases, you lose some of the cells, you have some impairment, but mostly what happens is you just follow these dispersed speckles. Now, there's another drug called cantharidin that can actually allow them to fuse. Oh, this has moved, sorry. So you can see these larger speckles forming. Now we're going to treat the cells with ICM to give rise to the CTCF clusters. And again, they nicely follow the new elongated form of these speckles. Does this mean that they mix? Does this mean that the CTCF clusters are actually embedded sort of inside the speckles, which one, biophysically would probably not expect because of the properties that these proteins have and the properties that speckles also have? Now, if you look carefully at what happens at ICM, you have the formation of these very nice CTCF foci at speckles, right? So now I'm going to treat them again with hexandiol, and you can see that you don't have a full alleviation of the speckles, but you have an almost full alleviation of the foci, the CTCF clusters. And what we went and did to make sure that our speculation was correct, we imaged these mixed signals that under the the confocal microscope or the wide field microscope appears absolutely colocalizing stuff on a STED super resolution microscope. And if you look at the large speckles and large CTCF clusters, you can't really see much. For example, you can see here that this large cluster has two little things on it, and it's not the whole thing covered. But if you really look into the small ones, you can clearly see that in red you have a speckle, and like a little hat, the CTCF cluster seems to be sitting like this on top of it. So we really think that these are not mixing. If anything, these are sort of coalescing, or maybe just two different blobs, one having some sort of local attraction for the other. And then what I asked Spiros, who is the PhD student that was doing all this work, to do was to do the following experiment. I said to him, well, okay, if this is all phase separation and stuff, maybe what we can do is we can overexpress one of these unstructured domains of SRM2 inside the cell to see what happens. And he said to me, well, that's a great idea, boss, but there's a bit of a problem. These are very, very repetitive and very large, and they are impossible to clone. So what we managed to clone in the end was a rather small piece that I think is around 300 base pairs that is unstructured, but also contains the RNA binding domain, the mapped RNA binding domain of this large protein. So what we did is we cloned this into a piggyback transposon, and we put it into the cells, and we can now turn this on at will with docks. So these are happy IMR90s before docks. You can see here speckles forming in every one of them. And now I'm going to turn it on in this cell, but not in the neighboring one. And you can see that when I do this, so these cells are young, right? They don't have CTCF clusters. When I do this, the excess RNA bind domain of SRM2 goes to these large foci in the nucleoplasm and to the nucleolus. And then when you look at what these foci are, they are SC35, so they are speckles. So it goes to existing speckles, and it draws with it a reasonable amount of CTCF. Although this is a young cell and shouldn't have any CTCF clusters. And what is very important is that CTCF does not get attracted to the nucleolus, despite the fact that SRM2 also goes there. Now, let's do the proper experiment. Let's remove SRM2 from the mix. So now we're going to knock down SRM2. If you do a scramble control, right, CTCF in senescent cells, they will form clusters and they will colocalize with SC35. If I knock down SRM2, no speckles, no CTCF foci. If I knock it down and overexpress just the RNA binding domain, just this very, very small part of this huge protein, it all comes back to normal. Perhaps the foci are slightly smaller and slightly more. This we haven't properly quantified, to be honest, but you can clearly see that this is enough. You see what it looks like. It also goes to the nucleolus. These are the speckles that form outside the nucleolus, and you see that the CTCF only clusters on the speckles. And you can actually, sorry, I didn't put it in, but you can actually do this also in the young cell. Take away the speckles, put this back, and it will draw CTCF again into the speckles in the absence, the full absence of SRM2, which I find incredibly impressive. This is all you need. Now, this, of course, could be that this is just an unstructured domain, right? So we take an equal-sized unstructured domain from the other side, the other IDR, that has a very similar, as far as we can tell, composition of amino acids. Definitely not the same relative placing, of course, which might play a role, but a very similar composition. We overexpress it. Here's the docs. You express here, but not there. And it will not go specifically to the speckles. It might go with the nucleolus, and there's no clustering being formed. So not any part of SRM2 will do it. Clearly, the RNA bind domain will, this will not. So we now understand how this happens and on which entity in the nucleoplasm it happens. The problem we have is that we cannot take the images that we look under the microscope at and put them on a high-C map and say, oh, these are the regions of the genome that actually go there. And we really try to do this. And I'm going to show you. So essentially, the next 4 or 5 slides are our futile attempt to understand which genomic regions are involved here. So what we did, because these were loops, we thought this would be CTCF loops. We said, okay, which technique would give you the most loops? And there's this variant of high-C called micro-C, where instead of cutting the genome with an enzyme, with a restriction and a nuclease, you're cutting with micrococcal nuclease that cuts very, very often, and apparently gives you these beautiful high-resolution maps where you have lots of loops being mapped. And this is a control cell. And this is a cell where we've added ICM. And you can appreciate from the positions of the new arrows that there are some new or stronger loops that occur in these cells. And if I treat with hexandiol, some of them will either weaken a lot or disappear, okay? So our initial thought was that the formation of these extra loops actually represent this large clustering that we see under the microscope. But you know, how can you say, well, how can I really say that if this region here or that region there or that region there is actually associated with the speckles? That's very difficult to do. So we approach this using a 3D genome modeling approach. So we're using Chrome 3D that was developed by Jonas Paulsen when he was working in Norway as a postdoc. And what this does is it makes only 2 assumptions to make models that look like this, that of 3D models of an individual chromosome. So the assumptions are the following. It uses TADs, so each sphere here is a topological domain, as the beads that make up a chromosome. And it uses a signal of high-C or micro-C in our cases to say how much do any 2 TADs interact. So the more they interact, the closer they're going to be on this polymer model. And then the other thing it uses to make, because you make a model of the whole genome, it uses information about lamin association. And because we were lucky, there's DAM ID from this particular cell type, we know which regions are more associated with the lamins and which are not. And using these 2 criteria, you can make models that look like this. And then after the model is formed, we actually go and paint red beads that we know, well, that we presume are associated with speckles, because we've done a cut and run of SON. So this is one of the key components of the speckles, and other people have shown that you can sort of find this association on chromatin. And if we get strong positive signal for SON in a given TAD, we will color it red for association with the speckles. There's, you know, or there's, sorry, pink and red. It doesn't matter. You treat them as the same. They're both associated. The topological domains more or less associated with speckles. And if you look at these red positions, they tend to be closer to one another, the red beads, than they are to gray beads. So there's a bit of clustering already. This is the structure of chromosome 11 from a young cell, deduced from a young cell. And now I'm going to show you what it looks like from an old cell. I think you can appreciate even by eye that it looks, well, a bit different, definitely more condensed, definitely with more red. And clearly the red looks more clustered to the eye. And this is true when we actually look at this. So when we look at the difference between the 2, you actually see a decreased average distance between any 2 red beads compared to the distance to any gray beads. Now, as a sanity check for this, what we've done is we've simply taken chromosome 14, for example, here, right? And we've switched. So this is the young chromosome 14. And if you put on it the SON association of the old, it will not cluster as much as the real association of the old or the young. So if you put a non-condition relevant data set, if you paint these beads according to that, you will not get as much clustering as if you were using the proper control. Now, are these models an approximation? Of course they are. Would the chromosome look exactly like that? Probably not. But they give you an idea that, these SON proximal regions, they are probably talking to one another to some extent, and they could presumably be the positions where you have the clustering. And now as the last thing, I want to show you what this would look like. So if we take one of these models, right, and this is chromosome 10, I'm going to draw your attention to these beads here. You see, you have these beads here, and those beads, they're marked all by a little blue dot, and they represent beads in this and in that region of our micro-C map. And this is the interaction between them. And now I'm going to show you what it looks like in the what it looks like in the old conditions, right, where the relative signal here is now less, because some of these beads that used to interact move away and now become SON associated, and they cluster together. And this is the result that we see within this domain here. So these beads here, they belong to this domain, and all of a sudden they cluster more together. And they cluster more with domains, with these beads there. So it seems as if we are able to use these models to make some first assumptions of which regions might be coming together in 3D space, and then using things like DNA fish also to try and really show that these are associated with the speckles and the CTCF clusters. So 6 are a senescence-specific genomic landmark. They associate with speckles. It's all nice and good. We now understand how they form. They do the things, they are an early commitment landmark. All these are fine, but there's many open questions that we need to still address. So for example, how would HBGB2 actually work to induce these 6 if it doesn't talk to CTCF? Speckles are really rich in RNA. Does RNA play any role in their formation? And of course, the most difficult of these questions, which would be, is there any functional purpose to them? Are they just a sort of a byproduct of a cell that has aged and things cluster randomly? Or is this an educated process by which the cell actually tries to do something? Now, all the work that I've shown you thus far is being led by a single PhD student who is, the last few days, apparently a doctor. He passed his Viva, Spiros. And the computational bits are being supported by Ilya and Athanasia. So there's 3 Greeks on this project. I'm just realizing as I'm saying this. And Mariano is a physicist, also from the southern bits of, Italian physicist from the southern bits of Europe, assisting with 3D modeling. And with that, I want to thank you all for bearing with me, and I'm happy to take any questions.
Thank you very much for that great talk. So we're gonna open for questions. Rosa, go ahead.
Hi, thank you. Thank you very much. It was an excellent and amazing talk. Very interesting.
Thank you.
I was wondering if you have measured what happens with the splicing if it slows down as these speckles form.
Yeah, so that is a very good question. So there are splicing changes between young cells and senescent cells. That's absolutely true, right? So the question is this why this is happening? And CTCF has been implicated with splicing. I'm not sure how we would be easily able to show this. So what we're now doing is we've done a knockdown of SRM2. So we've removed it from the mix, and we're asking what sort of splicing changes happen there. But this data is not analyzed yet, trying to correlate it to the splicing change. But there's lots of splicing changes that happen with age, for sure.
Okay, thank you.
Paula, go ahead.
I have a question, maybe out of ignorance, because I'm not working in this field, but I was wondering if these long CTCF loops are also like the reason why you have lower transcription rates. So if you can mimic those long loops, can you make a cell senescent and like decrease the transcription rate?
Yeah, no, this is an excellent point. And here is the the big trick of the whole thing. It's actually the other way around. The lower transcription is what allows longer loops to occur. And it took us a very long and sideways trip to realize that. So we were thinking, exactly like you suggested, that maybe these longer loops, they bring things together and they sort of trap them, and you don't have enough transcriptional output, and this would explain things. And as this project was developing in the background, we did another project where in a cancer cell line, because it was easier, we made a degron of RNA polymerase II, because we wanted to see if you remove the RNA polymerase II, do you have differences in 3D genome organization? And the major effect that you see when you remove RNA polymerase II is the emergence of long-range CTCF loops, significantly longer than what is in the genome. And the reason for these long loops is there's less competition with transcription. So the extrusion by cohesin is now effortless. It has fewer barriers along the way, and it's easier to extrude these very long loops that become very, very obvious in our Hi-C and micro-C data. So we are absolutely certain that the reason why we see these is a combination of losing HMGB2 and having less transcription. It could be that, because also losing HMGB2 means less transcription to begin with, right? This is the first thing we found. So it's actually the loss of transcription that leads to the loops and not vice versa.
Thank you.
Diana.
Sorry, my camera was a little bit slow. Oh, that was a great talk. Thank you very much. I have a few questions. You mentioned several drugs like tautomycin, CANT, and hexandione.
Mm-hmm.
So is it known what is the chemical basis for the production or reduction of speckles in terms of their structure?
So hexandiol is a drug that interferes with the hydrophobic interactions between the unstructured domain of proteins. This is what it's designed to do. And by interfering, it doesn't allow the phase separation to fully form. So you essentially disperse any sort of membraneless entity you have in the cell. And it doesn't just affect speckles. Hexandiol is really, really radical as a drug. It affects many, many things. And to be honest, there's a very nice paper from Kazu's lab in Japan, where they look at what the effect is using biophysics and looking at chromatin, what the effect is on the biophysical properties of chromatin. And if you put the hexandiol, chromatin starts behaving more solid-like and less liquid-like, just because you are removing this propensity. Now, the other two drugs that we use, tautomycin and cantharidin, that can affect the fusion and dispersal of speckles, these are small molecule inhibitors with very well-known targets. Both of them affect phosphatases, and mostly PP2A, which is a phosphatase that also dephosphorylates cohesin. And now whether the two are connected, I don't know. But it's a very well-known drug for many, many years that you can manipulate speckles with that, because somehow you need the phosphorylation-dephosphorylation cycles to be active in order to have a specific size. And this is also, again, known. Phase-separate entities, they can be modulated by phosphorylation levels. Sometimes in their favor, sometimes for their dispersal. So by changing the phosphatase, you're actually changing the shape of these. You allow them to either grow more or to start dispersing.
Okay, thank you.
Sure.
Thank you very much.
Andres?
Yes, thank you. Thank you. It was a really nice talk. I have a few questions. First of all, I was wondering if there are differences in post-translational modifications of CTCF between the proliferating and the senescent cells, but maybe that's why it becomes more likely to phase separate.
Yes, there are. And it's difficult to put our finger on it, but we do know that when you enter senescence or when you treat with ICM in the short term, you actually find a smaller isoform of CTCF, and this is not smaller because it's it's a different isoform. It has some exons more or less. It just has some different modification on it, and it's clear that that the emergence of this isoform allows the formation of these clusters. Now it is a question of what exactly it is and how do you manipulate it, but it's absolutely clear that this is needed. Wow,
that's really interesting.
Yeah, and we have there are candidate positions on CTCF that we are now trying to mutate to see if we can either induce this or not allow clusters of CTCF to ever form. Yeah.
Well, thank you.
Actually, an experiment in the incubator right now, as far as I know, where it's the first try for this. So, yeah.
Okay, well, hopefully it will give very nice results. So I have another question. I'm just curious if is there an increase in the number of speckles in senescence, or I'm just wondering if CTCF now moves to pre-existing speckles, or it can also form new ones.
So it's not the easiest thing to quantify these, and of course there's quite a bit of heterogeneity. Despite the fact that we use this drug, and this drug has all these nice effects, right? Everything is cleaner and more homogeneous. It's still not perfect. Our general impression is the following: they tend to become more round and slightly larger in senescence than they were in the interphase, where they were somewhat smaller and more irregular. So you would find multiple different shapes. And actually, the longer you treat with the drug, the more round and obvious and large they become, which could sort of play with this idea that they may transition from a liquid-like state that is a bit more, wobbly to a more solid state, more gelated state, as people say. But this is sort of our observations with eye. We have not quantified that.
Okay, well, thank you. And I have just one last question.
Sure.
Have you seen CTCF now coming to proximity with another phase-separated condensate, the nucleolus, or?
We've never seen it in the nucleolus. Never. Even, you've seen these experiments. Even when we overexpress the RNA binding domain of SRM2, and it goes everywhere and it attracts CTCF, it cannot attract it into the nucleolus. I guess that environment, which there's a bit of debate right now whether it's actually phase separated as was initially, or whether it's because the nucleolus has structure. It has a nice mixed structure, but it has structure. But clearly, it's an environment that CTCF does not like going into, probably for reasons of charge or whatever.
Okay, well, thank you very much. It was really interesting.
Thank you, Andres.
Felix.
Hi, one more time. In the same sense of one of the Andres question, I was wondering, what is your opinion concerning the possibility that part of the cofactors allowing CTCF to generate this structure could be RNA?
Yes, I have a very strong opinion about this, actually.
Just recalling that it has relatively recently demonstrated that CTCF binds RNA, no?
Absolutely. So that was our idea at first. Mm-hmm. And what we did is we've done a number of things. So the first was to clip CTCF. So we did a clip to identify the RNAs it binds. And at face value, it binds way more RNAs in senescence than it does in the young cells. So we thought that was very nice, right? Oh, okay. But we have a piece of evidence that I can actually show you because it's on the next slide, which I find really fascinating, to be honest. So when you do a co-IP, so this is the co-IP that we do. So when you do a co-IP, you usually get rid of RNA and DNA using benzonase because you want to only identify direct interactions, right? So when we did our co-IPs for the mass spec, this is the conditions that we used. So here in this particular case, we pull down with SON, which is a key component of the speckles, and we blot for CTCF, and we find a band like the one we would expect. If you don't use benzonase, but use RNase instead, this interaction becomes 50 times stronger. So if anything, the RNA is working to make the interaction more separated, as if the RNA is the interface between the speckle and the CTCF cluster. And if you use DNase, so you remove all the DNase, but if this is RNase free, so the RNA is still intact, you get essentially no interaction.
Okay.
So my guess is that the speckle that is very RNA rich keeps the CTCF outside of its core via using RNA, and this is probably the RNA that we're capturing in the clip.
Okay. Nice, very nice. Thank you very much.
Okay, I don't see any more hands, so I'm gonna jump with one before another one raises their hands. So, well, just two questions. One, why do you think there is this different susceptibility to the PS inhibition in forming the clusters at an early or later stage of senescence?
The only thing I can say is that when we look at, again, just a simple Western blot with the isoforms of CTCF, in the early stage, we see the formation of the smaller isoforms. In the later stage, they disappear again.
Right.
So I think that maybe the induction phase has all these components that, somehow the drug can go in and disperse them. But then when they transition, these are not necessary anymore for whatever reason.
Right.
And this is the only evidence that we have.
Okay. And just one more. So you showed us this coincidence of CTCF and HMGB2 at boundaries in the meta plots, but then really in the chip and in your experiments, they really, I mean, even in the chip, the binding of HMGB2 is more like you said, no, like obstructing the loop formation, and also in the colocalization, they seem like together but not mixing, let's say.
Yeah.
So what's your idea behind what could be doing as a boundary or kind of nearish CTCF, or just to kind of think on those two results together?
So my favorite answer to this is the following. If you go back to the Dixon paper in 2012 for the TADs, 20-something% of all TAD boundaries they identify are actually active promoters rather than CTCF. Of course, CTCF is at the base of most loops, especially the structural loops. But when it comes to TAD boundaries, CTCF is not at the majority. So then you ask, where does HMGB2 bind? And it binds almost exclusively at promoters. It has some positions either in introns or outside, but the vast majority of peaks is at promoters. So to me, this means that the enrichment of these peaks are enrichment of active promoters at or very close to the boundary. So they might have a CTCF a bit to the left or to the right, but they're never on it because these are promoters and they're not boundary CTCFs. Now the question is that enough to sort of stop the CTCF from looping to another CTCF? You could view it as a sort of boundary kind of element that we know promoters have as a function to some extent to extrusion and formation of loops. And this is why I say that when you reduce transcription, so essentially when these promoters get tuned down or deactivated, then you can have better extrusion. You can now see these longer loops. I think this is how I explain it. So it's not a direct thing. It doesn't involve one talking to the other.
Right. Okay, thank you. Rosa?
Thank you. It's me again.
Hi.
I was wondering, it just caught my attention that you said that with the senescence, these speckles are rounder, and that reminds me that usually the phase separation in cytoplasm as the cell ages, they become more solid. Those granules become more solid, and it's difficult to break them with hexadiol or something like that. So it, as you were explaining, that they were growing with age and they were more round, and more round means like that they are liquid, more liquid, because when they become solid, they are more like fibrous.
Yes, that is true.
So I was wondering whether it's actually protecting so that the splicing can still go on instead of trapping some splicing elements, so it doesn't work anymore.
So we want to think that they are not a byproduct, a random aggregate of senescence, but that they have a function to serve. And of course, we want to think that because it makes them more interesting. And I do think that this is what they do, exactly what you described. I think that you are bringing via CTCF a selection of chromatin loci close to the speckles to make sure that they are spliced better. And that's why we've done the SRM2 knockdown in senescent cells to see the splicing of which genes is going to be affected, and then see whether these genes are likely candidates to be actually associated with speckles. And this is exactly my speculation, that you keep the few genes, because these cells are senescent, but they're very metabolically active, right? They're still alive. So you keep the essential stuff close to speckles to make sure that at least these get spliced and transcribed, and I get to live another day.
Yes, exactly, because it's the contrary in the cytoplasm, where the things are just getting really solid and they are not round anymore. These granules are not round and they are like fibrous. And I was wondering, how much hexadiol did you use?
So we use, and of course I'm going to get this wrong, but it's a ridiculously low amount. So we treat for 1 minute.
5% maybe?
I think it's 6% for 1 minute.
Okay, because I was going to suggest that you should play with the amount of hexadiol. So you take the where you have the speckles, and then you move to different dilutions of hexadiol, and then you move to the one that doesn't dilute them anymore. They are still in the control, they are there. And then do you use that?
Yes.
The same amount for the others that are forming with senescence. And sometimes they don't dissolve, they dissolve if they are like softer.
Yeah, exactly. But that's what we have. So with the 1 minute, you dissolve few of the speckles, but you dissolve most of the CTCF clusters.
Okay.
The other thing that we've tried is to use longer times to try and dissolve the late speckles that we think are not solid, but they're probably more gel-like. And even if we go up to 3 minutes of 6%, we don't see any effect. Now, we haven't played that much with it. I mean, it's a very nasty chemical, and these are primary cells, so they're very, very sort of unhappy if you overdo it. But in that range, we don't see an effect.
Okay, great.
Okay, well, I think that would be all. So let me just thanks again, Argyris, for the very nice talk. I think from many of us it was really, really interesting, and we will try to bring you again, maybe live, so that you can actually get to know Mexico City. And
I'd love to, of course.
Excellent. Okay, well, thank you very much, everyone.
Thank you, everyone. Yes, and Mayra, thank you very much for the invitation, and Andres for the discussion before the talk. And thank you, everyone, for attending. It was a real pleasure.
Thank you.
Thank you.
Bueno, buenas vacaciones a todos. Good holiday.
Igualmente a todos.
Igualmente. Hasta
Yes.
luego. Bye bye.
Ciao.